name is John Nyagaka. I'm going to present my CS thesis that is on smart predictive analysis in clinical trial research, the case of coronavirus COVID-19. The coronavirus has presented us with an unprecedented threat, an unprecedented opportunity to come together as one against a common enemy, an enemy against humanity. COVID-19 has wreaked havoc, and its impact has been both devastating and catastrophic. The disease's origin is believed to be in Wuhan, China, and it was first discovered in late December 2019. It has since penetrated and ravaged every country of the world. As of to date, the number of infections stands at 158 million people. Among these, over 135 million people have recovered, representing 88% of the total infections, while 3.2 million people have sadly lost their lives, representing 0.02% of the total infections. The remaining percentage, 11.9%, represents active cases. Even with the percentage of deaths seemingly negligible, loss of life is a huge loss to productive members of society. Hence, there is need to mitigate this pandemic, and one way is through vaccine development through clinical trials. Clinical trials are a way to test new methods of diagnosing, treating, or preventing health conditions. The goal is to determine whether something is both safe and effective. Clinical trial research on COVID-19 presently comprises of three categories. 1. Ongoing clinical trials for non-COVID drugs. 2. Clinical trials for repurposing drugs to treat COVID-19. And 3. Clinical trials for new drugs to treat COVID-19. Vaccine development is inevitably a multi-stage and multifaceted process. Currently, several vaccines are under development, and hence clinical trials research is ongoing. Clinical trials are done in phases, which take months and sometimes up to years. There are 4 critical phases in clinical trial research. 1 is research and development. 2 is clinical development. 3 is manufacturing and supply chain. And 4 is launch and commercial. Patient recruitment falls under the first phase of drug development process, that is research and development. Recent statistics from Deloitte indicate that patient recruitment is one of the highest cost drivers in the vaccine development process, accounting for 32% of the cost. This has been attributed to inefficiencies occasioned by traditional methods used in the process, calling for the need to develop efficient methods to make the process seamless. This is one of the gaps identified in the process. Among other gaps identified is the inconsistency of randomized clinical trials that are often misleading due to the complexities associated with the first phase of drug development, that is patient recruitment. Also, the Journal of Clinical Epidemiology identifies artificial neural networks as being more efficient than traditional logistic regression methods in the clinical trial research process and proposes the need for them to be adopted. Different solutions have been developed. However, recent emerging technologies in clinical trial research are centered in 3 critical applications, that is, first, clinical trial design, 2, clinical trial optimization, and 3, patient recruitment. In this regard, several products have been developed to mitigate the challenges occasioned by inefficacy in clinical trial research. These include 1, cont'd, which uses algorithms trained on high volumes of patient eligibility criteria for clinical trials, and thousands of open trials are searched by the cont'd match search engine based on user responses to a questionnaire. And secondly is Deep6 AI, which uses NLP, natural language processing. Deep6's algorithm are trained on patient medical data and thousands of clinical data points, such as symptoms, diagnosis, and test results, and they are extracted from patient health records. These solutions have not explored the use of artificial neural networks in predicting the eligibility of patients in electronic health records for a vaccine trial, which is the gap this research seeks to concentrate on. The problem statement deduces that the clinical trial research and design requires a lot of precision, and so does one of its critical phases, patient recruitment, hence the need for its efficiency. With reference to the problem statement, the research objective, therefore, is to significantly help in improving the efficacy and efficiency of clinical trial research and design by improving the patient recruitment process. While the hypothesis indicates that deep neural networks can be used to match patients who fit a drug development agency's recruitment criterion efficiently, the research will seek to answer the question on how effective could artificial intelligence methods be in matching patients who fit a drug development agency's patient recruitment criterion in the development of vaccines for COVID-19. Seeking to meet the research objective, evaluate the hypothesis, and answer the research question, different methodologies were used. The datasets used for this research were patient precondition dataset that comprised of patient records with their respective morbidities and if they had been diagnosed or not, while the clinical studies dataset comprised of various vaccine trials for COVID-19 with the respective inclusion and exclusion criteria that were used to form the basis of eligibility for the patients. Other tasks involved data cleaning and validation, and then data sampling. Oversampling was done due to imbalance in the data, and the technique that was used was adaptive synthetic sampling, which oversamples the minority class by generating synthetic instances for it by considering the density distribution. The approach allows for flexibility in the changing decision boundaries for samples that are difficult to learn. A binary classifier was built as our model, and the activation functions used for the input and hidden layers was the rectified linear unit function, while the sigmoid function was used for the output layer. The loss function utilized was the binary cross entropy, while the optimization method utilized was the stochastic gradient descent. Training was done on 70% of the oversampled data, while 30% was used for testing, and training was done over 100 epochs. The model attained an accuracy of 98.24 after the hyperparameters were tuned. This accuracy was attained with a learning rate of 0.001, and the model accuracy for both the training and testing is as shown by the model accuracy graph below. This was the optimal point where the model did not either overfit or underfit, as indicated by the model loss graph below too. The graph shows how validation and training loss decrease evenly and smoothly. Even with this accuracy, our model needed to be evaluated further. Other metrics used to evaluate the model include the confusion matrix. As per the confusion matrix below, the binary classifier predicted that out of 15,617 patients, 841 were eligible for a COVID-19 clinical trial, while 14,776 were not eligible for the same. In reality, 566 patients were eligible, while 15,051 were not eligible. As per our model, 14,776 patients are true negative, while 275 patients are false negative. Our model did not have patients who fall under false negative, while 566 patients were true positive. The misclassification rate is 0.0176%, and it indicates how often the classifier is wrong, while the recall rate is 0.98, and it indicates how often the classifier is predicting yes when it is actually yes, and the precision rate is 1, and it indicates how often the classifier is correct when it predicts yes. With these metrics, we were confident that our model had performed well. The binary classifier achieved an accuracy of 98.24, and other metrics like the confusion matrix affirmed the high efficiency and accuracy of our model, and this further affirmed our hypothesis. The constraints that we faced is that our dataset was quite small, and ideally deep neural networks perform better where we have large datasets. Patient health records are highly confidential, and sometimes access to them is quite limited. And future work recommended for this research involves improving the model to incorporate inductive transfer learning, where the model can utilize previously existing learning tasks and apply them to newer tasks, and then building a model that integrates both deep neural networks and natural language processing to use both structured and unstructured datasets, that is the electronic health records and textual data. Thank you.